Hello, my topic is imbalance in the helper 17 cells and targeted therapy in an infant with SAN-like syndrome. Definition: Th17 cells are a type of cooperating T lymphocytes responsible for eliminating certain pathogens, mainly fungi and mycobacterium that cannot be eliminated by a Th1 and Th2 cellular response. Meconium aspiration syndrome (SAM) occurs when babies inhale meconium into their lungs. During or after delivery, meconium aspiration is a form of neonatal acute respiratory distress produced by aspiration of meconium-stained fluid. Application in nursing. AVLAF treatments targeting Th17 cells include biologic drugs that are currently licensed for psoriasis therapy, such as ustekinumab and secukinumab. Ustekinumab was recently evaluated with encouraging results in 2 patients with DSP mutation, but we had obtained little kinetics improvement in another patient with SAM syndrome. You has DC1 mutation.
Th17 cells in particular are very interesting because they're part of our adaptive immune system. So they have a protective function where they protect us from fungal infections and extracellular bacteria. But at the same time, they can actually cause serious disease. They can turn against us and make us sick in the sense that they're heavily implicated in autoimmune diseases. We had studied Th17 cells previously at the population level, and it became clear that despite we could learn a lot, that at any given time the population was very heterogeneous. So they were all called Th17 cells, but they weren't exactly the same thing. I would say single-cell RNA-seq allowed us to look at single cells, but then at a genome-wide scale. So previously, what you could do at the single-cell level was look at maybe a handful of markers that you would know of, but you would have to know which genes you would want to detect. When you do single-cell RNA-seq, you actually get you're assaying the whole transcriptome. So you're looking at all the genes being expressed in individual cells. It allows you to characterize cellular states because you're looking across the whole populations, and you can get an idea of what type of cells are present in a population that you thought previously were basically the same based on those 5 markers that you were looking at. So another great advantage of single-cell data is that you can now look at gene expression covariation within single cells. That means that you can look at, given that I know that this gene is implicated in a pro-inflammatory response, you can start looking at other genes. Are they usually also upregulated when this gene is upregulated, or maybe they're always downregulated when this gene is downregulated? So you can start assuming that these genes, if they move together or move oppositely, they're sort of biologically bound in function. And that allowed us to prioritize genes to go after in terms of functional validations, and that might be implicated in the pro-inflammatory behavior of pathogenic Th17 cells.
The CD5L story turned out to be really exciting. So from the single-cell story, we've found that CD5 could be potentially a regulator of the different functions of the T cells. And in fact, what we showed is using the autoimmune disease model in EAE, we actually showed that this is a major functional switch. So if Th17 cells without this molecule, they suddenly can turn on the pathogenicity and cause disease. What we already know from the literature is that CD5L is actually an inhibitor of a fatty acid synthesis pathway. What that means is it actually inhibits the making of lipids and fatty acid inside the cell. So from this angle, we decided to ask the question, really, what are the lipids that are being made in the T cells or being transported in, transported out? And so we did a comparison of non-pathogenic versus pathogenic Th17 cells and found this really amazing correlation of the lipid profile of these cells and their function. And so from that angle, we started asking the question, well, does this lipid actually contribute the function? And we led to the conclusion eventually that depend on how much of the saturated fatty acids versus the unsaturated fatty acids you have, or polyunsaturated fatty acids you have, you can actually now deter the functional fate of these Th17 cells. And in fact, depending on what kind of fatty acids you have, either saturated or polyunsaturated, you can decide whether the RR gamma T is going to turn on genes that are in the pro-inflammatory program or turn on the regulatory program. So this really would open up the field about addressing the question of, can you influence a disease status and the T cell function by changing the environment that involves lipid, for example. The other application comes from Th17 cell itself, in that it's being driven by several transcription factors such as RR gamma T, which really direct the cell towards particular lineage. And so my finding actually showed the lipid can specifically regulate this transcription factor towards different functions. It doesn't just inhibit or promote, but it can actually change its selection on what genes to turn on or turn off.
So I think the strength of this is that you can actually not just look at general Th17 differentiations or maintenance, but you can go after a specific process. So in our case, that's Th17 pathogenicity. And if you can figure out what regulates Th17 pathogenicity, you can also start figuring out how you might tackle it, how you might prevent it.
Pros and cons. Pros. If treat the early meconial aspiration syndrome as positive prognosis and does not have to present further complication as delivery, the specialist can check for the presence of meconium in the baby and in the immunity fluid. However, before the problem can be detected, B. Measuring the heart rate through fetal monitoring, one of tests that are done to detect this type of problems and measure the health of newborns in general is the Apgar test. Cons. Chemical aspiration, pneumonia, brain damage due to lack of oxygen, shortness of breath lasting severe days, pulmonary atelectasis, persistent pulmonary hypertension of newborn. ASHU apply in Ecuador. Between 9 and 50% of the participants are complicated by the presence of meconium in amniotic fluid, and historically 5 to 12% of those cases develop SAN. This figure has dropped in developed countries to percentages between 0.6 to 3.9%, considering data and SAN incidence of around 0.3 93% of all live births, because 99% of the Th17 cells in our patients had a memory phenotype. We hypothesized that land survival memory, the Th17 cells, will not be vulnerable to the neuralization of interleukin. 23 where secukinumab could neutralize the effect of the perforate Th17 pool by blocking the action of interleukin 78 on the skin. Conclusion: the major effect of secukinumab in our patients' quality of life support interleukin 17A as a new target to treat patients with serious skin disorders. Treat will also join this group of cells that are both collaboratable and regulatorily, since in addition to their non-suppressive, they will also they will also coordinate the first steps of viral infection directly as well as indirectly induced by securing TGF-beta, the differentiation of the H17.
And then effector function. And this is kind of one of the points where I think the professor that I got a lot of this information from and in the textbook were not necessarily agreeing with each other with regards to the cytokines that play a role in differentiation. Keep in mind that at this point we're really starting to kind of diverge into what we think we know and then what we actually know. But what we know for sure is that interleukin 23, or at least for the purposes of this class, interleukin 23 is the cytokine that plays a role in Th17 differentiation, which results in the production of the transcription factor known as ROR gamma T. It's a TF for transcription factor. And then for the hypothesized ones, I'm just going to do those in gold and put a question mark by them. So for the ones we're hypothesizing is interleukin 16 and then transforming growth factor beta. And what we think happens from there is that we end up with production of interleukin 21 that is acting as an autocrine, which results in the production of the STAT3 transcription factor. This is not perfectly well known, whereas this is something that at least my PI was really stressing. So as for the effector functions, what we're doing here is we're really focusing on fighting extracellular pathogens. And keep in mind with both differentiation, the differentiation is reversible, and then the effector function is basically determined by the cytokines that we make after contact. I'm not going to write that out because it becomes redundant after this many videos, but the predominant, I guess, cytokine involved in the effector function is interleukin 17. Interleukin 17 is going to act on the stromal cells and then epithelial cells and cause them to start to secrete CXCL8, which if you remember back, I think when I did a neutrophil homing video, CXCL8 is a very common chemokine. And that doesn't really just also enhances neutrophils, but also to a lesser extent enhances macrophages as well.